Hi, this is Dr. Annie Gomes. I'm dealing with the paper Molecular Biology and Genetic Engineering. Today's unit is Application of Genetic Engineering, and the module name is Edible Vaccines and Industrial Enzymes, that is, proteases and lipases. In this module, the outline is the introduction of edible vaccines and their attributes, defining, developing, and mechanism of action of edible vaccines, advantages, disadvantages of edible vaccines, applications of industrial enzymes, that is, proteases and lipases. At the end of the session, you should be able to define edible vaccines, understand the making and mechanism of action of edible vaccines, summarize the advantages and disadvantages of edible vaccines, and relate the use of enzymes, proteases, and lipases in industry. To start with, what is a vaccine? A vaccine is a biological preparation that improves immunity to a particular disease. It contains an agent that resembles a disease causing microorganism and is often made from weakened or killed cells of the microbe, its toxins, or one of its surface proteins. The agent stimulates the body immune system to recognize the foreign antigen and destroy it. It was first prepared by Edward Jenner in 1796 for the protection of human beings from smallpox, and later Jenner's work was continued by Sir Louis Pasteur. Now, what are edible vaccines? It is a pathogen protein. In this, a pathogen protein gene is cloned, and this gene is inserted into DNA of the plant. You can take potato, bananas, or tomatoes. Human beings will eat this plant, the body will produce antibodies against the pathogen protein, and humans are thereby immunized against these pathogens. Few examples of diseases controlled by edible vaccines are diarrhea, hepatitis B, and measles. As Hippocrates said, let thy food be thy medicine, is the concept behind adopting edible vaccines. What is this concept? The concept of edible vaccines was developed by Adson in 1990. In this, introduced genes of interest into plants, genes expressed in the plant tissue's edible parts. Genes encode protective vaccine antigens from viral, bacterial, and parasitic pathogens that cause diseases in human beings. These transgenic plants are used as vaccine production systems. What are the ideal properties of edible vaccines? They are non toxic, non pathogenic, and have very low levels of side effects. They should not cause problems in individuals with impaired immune system. They have long lasting humoral and cellular immunities. Vaccination is simple and it does not contaminate the environment and should be effective and affordable. These are the some properties we see in edible vaccines. Now, why edible vaccines? They are affordable. Okay, they do not need any purification. They are easy to administer. They are cost effective. No transport expenses if local crops are engineered. They're easy to store. They are heat stable, no cold chain maintenance, and socio-culturally readily acceptable vaccines for their delivery systems. Oral vaccinations, like the edible vaccines, which I mean to say here, have mucosal immunity at various sites by secreting antibodies. They do not, there is no worry of reuse, misuse, or lack of sterilization, and so they have a very low risk of infection. Immunization through DNA vaccines is an alternative, but this approach is expensive. As I've already said, edible vaccines are cost effective and the cost is reduced more if the transport cost is reduced if the local crops are genetically engineered. Going to the mechanism of action, the goal of oral vaccination is to stimulate the mucosal and systemic immunity against the pathogen. Edible vaccines, when taken orally, undergoes mastication process in the mouth and then total cell degradation at the intestinal level. There are some patches which are called as Payer's patches, PP as I have referred to them here in the PowerPoint. These are small masses of lymphatic tissues found throughout the ileum region of the small intestines. They monitor the entry of pathogens and regulate by secreting IgA, producing plasma cells. The mechanism occurs this way. There is breakdown of edible vaccine near the Peyer's patches on the outer surface of intestines, and these contain follicles. These follicles act as sites from which antigen penetrates the intestinal epithelium. The antigen then comes in contact with the M cell. The M cell passes the antigen to macrophages and B cell. The B cell is activated by the T cell, which provide immune response. The activated B cells produce antibodies, and in this way, immunity is activated by the edible vaccines. You can see this in this diagram. Here we see the antigens from vaccine near the Peyer's patches. They enter the M cell. The M cell passes them to macrophages and B cells simultaneously. The macrophages pass them to the T cells, which stimulate the B cells to produce antibodies. The activated B cell starts releasing the antibodies and the immune response starts. So, that is the initial immune response. How do we develop these edible vaccines? There are two ways. One in which the entire structural gene is inserted into the plant transformation vector and it is multiplied, transformed into the plant cells. In the second case, the epitope within the antigen are unidentified. It may be a DNA fragment or a protein, coat protein, which is inserted into a coat protein of a virus such as tobacco mosaic virus or cytomegalovirus, CMV. The recombinant virus is then used to infect stabilized plants. The resultant edible plant vaccines are utilized for further immunological studies. The production of edible vaccine is seen here. You take the isolated desired gene from the human pathogens. You have a vector plasmid which is recombinantly made with the RDA technology. Then you transfer it into the plant cells. This recombinant plasmid is transferred into the plant cells through the potato leaf here you can see, and from there whatever plants grow through the callus culturing, you have the transgenic gene from the virus, and that comes into the tubers. So tubers when administered raw can trigger the immune response system. You can see in the next picture, wherein you can see Agrobacterium tumefaciens cell, bacterial cell, which is used to infect the plant cell. The plant cells are then callus cultured, and from where whatever seedlings, the transgenic plants which will produce potatoes, which can be used as edible vaccines. What are the advantages of edible vaccines? Let's start. They storage near the site of use. They are heat stable, eliminating the need for refrigeration. These therapeutic proteins are free of pathogens and toxins, economical in mass production and transportation. Antigen protection through bioencapsulation and improved safety can be provided for them. Delivery of multiple agents can be done through them, and there is reduced need for medical personnel and sterile injection conditions. Generation of systemic and mucosal immunity is guaranteed here, and there is enhanced compliance. But there are some disadvantages also, like development of immunotolerance to vaccine peptides or proteins, consistency of dosage from fruit to fruit, plant to plant, or generation to generation is not similar. Stability of vaccine in fruit is not known. Then evaluating dosage requirement is tedious, and selection of best plant is difficult to be developed into an edible vaccine. So these are some of the disadvantages. Going to protease enzyme, which is the next subtopic. Proteases are enzymes which catalyze the hydrolysis of peptide bonds present in proteins and polypeptides. They are widely used in detergents, pharmaceuticals, followed by food industry. They find applications in leather processing as well as bioremediation processes. Proteases can be classified on the basis of their origin, catalytic activity, nature of reactive groups in the catalytic site. The major sources of protease enzymes are plants, animals, and microorganisms, both bacteria and fungi. They are grouped into two types. There are exopeptidases, endopeptidases, based on the site of their action on polypeptide chains. Usually, exopeptidases, they act on the ends of the polypeptide chain, whereas endopeptidases act randomly. Some of the plant proteases include bromelain, physin, papain, which are widely used in food industry for brewing, tenderizing of meat, coagulation of milk, and digestive aids. Proteases also help to improve flavor, nutritional value, solubility, and digestibility of food products and proteins, as well as to modify their functional properties, including coagulation, emulsification, etc. They are also widely used in baking industry for the production of bread, baked fruits, crackers, waffles, etc. These enzymes are used to reduce mixing time, decrease dough consistency, and uniformity. They also regulate the gluten strength in bread and help to improve the texture and flavor. Some of the proteases which are isolated from some species of Aspergillus has been successfully used for improvement of functional properties of wheat gluten. The addition of proteases could release sufficient peptides and amino acid levels in the wort to get proper fermentation. Acidic fungal proteases are used in improving fermentation of beer as they are efficient even at low pH by balancing the amino acid profile of the beer. They are also used in dairy. This application is associated with the improved flavor of cheese. Okay, they are used for the acceleration of cheese ripening to modify the functional properties and to reduce the allergenic properties of milk products. In making cheese, proteases are used to hydrolyze the specific peptide bonds to generate para-casein and macropeptides. Now, coming to lipases, lipases are a class of enzymes which catalyze the hydrolysis of long chain triglycerides. They are naturally present in the stomach and pancreas of human beings and other animal species in order to digest fats and lipids. Microbial lipases are produced by bacteria, fungi, and yeast. This enzyme finds application in various industries, including food, biofuel, detergents, and animal feed. It is also used in leather, textiles, and paper processing applications. In the food and beverage industry, lipases find major applications in dairy, baking, fruit juice, beer, and wine industries. They're also widely used in dairy for flavor development in dairy products and processing other foods containing fats. They can improve the characteristic flavor of cheese by acting on the milk fats to produce free fatty acids after hydrolysis. Different types of cheeses which are made using lipases include Romano cheese using keyed lamb regrastic lipase, camembert cheese using lipase from Penicillium camemberti, it's a fungi, and cheddar cheese using Aspergillus niger or A. oryzae, Aspergillus oryzae. So these are also fungi. Lipases catalysis could improve the texture and softness of cheese, as I've already said. They are also used to better the flavor of butter, margarine, to prolong the shelf life of a lot of baking products, and to improve the quality of cocoa butter. They find applications in ice creams, single cell protein, carbohydrate esters, and amino acid derivatives. Lipases are also used as biosensors in food industry. Immobilized lipase was successfully used for the determination of organophosphorus pesticides with a surface acoustic wave impedance sensor by lipase hydrolysis. It may be also used in the determination of triglycerides and blood cholesterol. In alcoholic beverages such as wines, aroma can be modified using lipase enzymes. In addition to this, lipases could also be used in the processing of different waste streams that are released from the food industries. You can, for your additional this, you can refer this references and additional some websites. Okay. I hope you have benefited from this module. Thank you.